Right now, in research labs all over the world, scientists might be, actively curing cancer.
Oh, absolutely. It's happening every day.
Right. They are pipetting these brilliant, incredibly complex chemical compounds onto tumor cells, and the cancer is dying. But there is a terrifying catch to this massive success, and that catch is that the cure they just found might only work on the specific sterile plastic lab dish it was grown in.
Yeah, that's the tragedy of it.
When doctors take that exact same miracle drug and give it to a human patient, it just completely fails. Why? Because the map we're using to navigate human biology is, frankly, fundamentally broken.
It really is. It is one of the most frustrating bottlenecks in modern medicine. The therapeutic landscape for oncology is just incredibly murky. I mean, we're dealing with diseases that mutate rapidly, but ethically and, well, medically, you simply cannot use a human patient as a testing ground for a dozen different highly toxic chemical combinations just to see what sticks.
No, of course not. You'd kill the patient before you cured them.
Exactly. You need a proxy. You need something else to test the drugs on first.
Which is where those lab dishes come in. To protect you, the patient, scientists use human tumor-derived cell lines. They extract cancer cells, put them in vitro, and run their drug trials there. And I mean, on paper, these cell lines sound like a perfect solution.
They sound like a dream.
Yeah. They are effectively eternal, they're cheap to maintain, and you can clone them endlessly. That means you can repeat a single drug experiment thousands of times under perfectly controlled conditions.
But, and this is a massive but, the problem with perfectly controlled conditions is that they just don't exist inside the human body. Growing cells in an artificial lab environment fundamentally alters their underlying biology.
Because they adapt, right?
Right. Over time, they undergo genetic and physical variations just to survive in the plastic dish. It's evolution on a micro scale. The cancer adapts to the lab, not the human.
So you end up with the ultimate false positive. You cure the lab-adapted version of the cancer, not the patient's actual disease.
And that disconnect, that exact gap, is the central focus of the really fascinating computational biology paper we are diving into today. A team of researchers, Benabithi Bose and Serdar Bozdag, have developed a groundbreaking new pipeline called CTD-PathSim 2.0.
CTD-PathSim 2.0. Catchy name.
Right. But you can basically think of it as a mathematically rigorous matchmaking service. Its sole mission is to comb through massive, and I mean massive amounts of data, to find the absolute perfect biologically identical lab-grown cell line to act as a proxy for an individual patient's unique tumor.
Okay, let's unpack this. We are talking about true preclinical personalized medicine here.
Yeah.
To really grasp how this new pipeline pulls off this matchmaking feat, we have to look at why older tools missed the mark. Because, scientists have tried to do this before with older models, right?
Oh, many times. There were tools like transcriptome correlation, the tissue similarity index, and Cellular.
And all of those older models suffered from a major blind spot related to something researchers call bulk. So for those listening, here's our first big concept to pin down. What do they mean by bulk?
So previous computational alignment methods essentially relied on a single omics approach.
Meaning they only looked at one layer of data?
Exactly. Usually just gene expression. They would take a tumor biopsy, grind it up, and analyze it as if every single cell inside that sample was an identical cancer cell. They treated the tumor like a uniform lump of bad cells.
But anyone who has looked at actual biology knows a tumor in a human body is heavily heterogeneous. It's not a solid lump of identical clones.
Oh.
It's a chaotic, diverse ecosystem, right?
It's incredibly chaotic. It is packed with cancer cells, sure, but it's also packed with the patient's own immune cells fighting the tumor, or sometimes, being co-opted by it to help the tumor grow.
You've got B cells, natural killer cells, CD4 plus T cells, monocytes.
Right, and ignoring that ecosystem completely ruins the drug trial. This raises an important question, right? Why does it matter? Well, the source material highlights a very stark clinical reality. Chemotherapy drugs don't just kill cancer. They profoundly affect a patient's immune cells, too.
Oh, wow. Okay.
Yeah. Cyclophosphamide, for example, is a heavy-hitting chemotherapy drug. It has been shown in clinical trials to drastically reduce T cells in cancer patients. Another common drug regimen for testicular cancer wildly alters natural killer cell levels for entire treatment cycles.
Okay, let me make sure I'm wrapping my head around the scale of this error. It's like we've been evaluating the health of an entire forest by only looking at the pine trees.
That's a great way to look at it.
We completely ignored the soil quality, the animals, the underbrush, the invasive vines. And if your lab dish cell line doesn't have the biological architecture to account for those specific immune cells, like the T cells you mentioned, testing a drug like cyclophosphamide on it is worse than useless.
Because you are measuring a reaction that physically cannot happen in the dish. There are no T cells in that dish to react.
You're measuring a fantasy.
Exactly, you're measuring a fantasy. Which brings us to how ctDPathSim 2.0 actually fixes this. If treating the tumor as one uniform lump of Praintree fails, we have to move from a single omics approach to a multi-omics approach. We have to map the entire forest.
Okay, so if you are listening and keeping track, here's a quick recap of our first 5 minutes. The old way of finding a cancer proxy failed because it treated tumors like a uniform bulk material, ignoring the immune system completely. The new mission is to map the whole ecosystem. But how on earth is a computer supposed to untangle those roots and vines? How does it map an ecosystem that complex?
Well, it starts by expanding the data pool. Instead of just looking at gene expression, the pipeline integrates 3 massive, distinct data sets to build a complete profile. You can remember it using a simple mnemonic, the MEC protocol. MEC.
MEC. Okay, I'm writing this down. What does that stand for?
M is for DNA methylation, E is for gene expression, and C is for copy number aberrations, or CNAs.
Let's spend a second on those, because that is a lot of dense molecular biology. So gene expression is the old standard. That's the E. It's basically looking at which proteins the cell is actively making, right? It's the lights that are currently turned on in the house.
Perfect analogy, yes.
But what does adding DNA methylation, the M, actually tell us?
So DNA methylation gets into epigenetics. These are microscopic chemical tags attached to the DNA itself. They don't change your core genetic code, but they act like dimmer switches.
Oh, I see.
They dictate whether a specific gene should be turned on, turned off, or dialed halfway down. A tumor might have the exact same genetic code as a healthy cell, but the methylation tags are completely scrambled, telling the cell to multiply uncontrollably.
So you have the lights that are on, which is expression, and the dimmer switch is controlling them, which is methylation, and the C, the CNAs, copy number aberrations.
Those are structural, physical changes to the genome itself. Imagine whole sections of a chromosome being entirely deleted or accidentally duplicated 3 times over. This isn't just flipping a switch. This is tearing down a wall or building an unauthorized extension on the house.
Right, okay. So by combining the switches, the active lights, and the physical blueprints of the house, the tool gets a highly holistic profile. But I'm stuck on a mechanical problem here.
What's that?
The researchers have all this rich multi-omics data, which is great, but it is still extracted from a bulk tumor biopsy. The forest is still all mixed together in the blender when they test the sample.
It is, and that is exactly why the pipeline uses a mathematical technique called quadratic programming to run a deconvolution algorithm.
Deconvolution. Okay, that sounds intensely sci-fi.
It sounds intimidating, but it's brilliant.
My understanding of deconvolution is basically unmixing things. It's like someone hands you a fully blended, completely pureed green smoothie, and this algorithm mathematically extracts the exact original proportions of strawberries, bananas, and spinach that went into the blender.
Yes, that is exactly what it does.
But how do you actually pull a pureed strawberry back out of a smoothie? I mean, once it's blended, it's physically gone.
Well, you don't pull the physical strawberry out. You look for its unique chemical signature. In our case, the algorithm takes the bulk tumor data and cross-references it against known pure reference profiles of specific amnere cells.
Oh, so they already have a cheat sheet of what pure cells look like.
Precisely. It knows exactly what the DNA methylation of an isolated B cell or a pure T cell looks like. So the algorithm hunts through the messy bulk data, looking for those specific mathematical anchors.
It's looking for the microscopic strawberry seeds floating in the blend.
Yes. To do this, it minimizes the Euclidean distance between the bulk profile and the known reference profiles using constrained optimization.
Okay, hold up. We are going to need to translate constrained optimization for those of us who haven't taken calculus recently. And Euclidean distance. That's just measuring the straight line between 2 points on a graph.
Exactly. You map the messy tumor data as one dot and the pure reference cell as another dot, and you measure the distance. And constrained optimization just means the algorithm forces the math to obey physical reality.
What do you mean by physical reality?
Well, you can't have -3 bananas in a smoothie. So the math forces all the variables to stay between 0 and 1, representing percentages, and it ensures they all add up to exactly 100% of the tumor. It mathematically filters out the noise of the bulk tissue to reveal the pure underlying immune and cancer signals.
That is staggering computational power. It mathematically unblends nature. Okay, so time for another quick recap for the listener. We've taken the bulk tumor, mapped its lights, switches, and blueprints using the MEC protocol, and unblended the smoothie using deconvolution.
We have our pure ingredients.
Right, but just knowing that you have 20% strawberries and 40% bananas doesn't tell you how they are interacting with each other in the body. Once we have the parts, how do we know what the cancer is actually doing with them?
We look at the biological pathways. In the next phase of the pipeline, the tool isolates very specific driver genes. It looks for differentially expressed, or DE genes, meaning genes that are significantly overactive or underactive. It also looks for differentially methylated genes and differentially aberrated genes.
The DE, DM, and DA genes. And reading the source material, they were incredibly strict about what counted as a driver gene. They weren't looking for minor fluctuations at all.
Not at all. For gene expression, the fold change had to be massive. For the structural aberrations, they only flagged high-level genetic amplifications or deep deletions. They only cared about the major architectural drivers of the cancer, not the background noise.
And once they isolate those heavy-hitting genes, they map them using international databases, right? Specifically, I saw they used the Reactome database and a curated list of 702 known cancer-driving genes from the COSMIC database.
Yes, the catalog of somatic mutations in cancer. And somatic just means mutations you acquire over your lifetime, not ones you inherit from your parents.
Got it. So they use those databases to see the communication networks. Because a pathway isn't just a list of genes, it's a supply chain. Are these genes driving the signal transduction pathway, which regulates how the tumor manipulates its environment? Or are they hijacking the immune system pathway to suppress inflammation so the cancer can hide?
Exactly. The tool maps these exact biological pathways for the human tumor sample, and then it does the same for thousands of lab-grown cell lines. Then it ranks how perfectly their supply chains match up.
Here is where it gets really interesting. The specificity this tool achieves is unreal. Let's look at the breast cancer example they provided in the paper. The tool analyzed a specific subtype, HER2 positive breast cancer, and it ranked a lab cell line called HTC-1599 incredibly high. It flagged it and basically said, yes, this lab cell is using the exact same biological supply chain as a primary HER2 positive tumor. Use this to test your drugs.
A perfect proxy.
But then it took another widely used cell line called UACC893 and ranked it at the absolute bottom. It gave a massive warning sign. That perfectly aligns with very recent clinical literature. Studies have shown that the UACC893 cell line has a weird quirk involving how it expresses the NET1 and NET2 genes.
Right.
Because of that quirk, the cell line behaves much more like a brain metastasis of breast cancer rather than a primary breast tumor. It mutated in the lab. It evolved to act like a cancer that has already spread to the brain. If a doctor tested a primary breast cancer drug on that cell line, the drug would likely fail because they're effectively trying to cure a brain tumor without knowing it. And the most brilliant part of all of this, the CTDPathSim 2.0 algorithm didn't read the clinical literature about NAT1 and NAT2. It didn't know the textbook answer beforehand. No, it just followed the pathway math of the multi-omics data, and it arrived at the exact biological truth completely on its own. Okay, but I have to play devil's advocate here. Biology is notoriously messy, and theoretical math in the computer model can look totally perfect on screen until it hits the real world. Did they actually test this against living patients taking real drugs? Because if this is just an exercise in making spreadsheets look pretty, it doesn't actually help anyone.
That's the most important question, and yes, they absolutely tested it, and they went massive with it. To validate this tool, the researchers analyzed 22 different cancer types, pulling data from over 7,200 patient tumor samples and comparing them against over 1,000 different cancer cell lines.
Wow.
Yeah. They generated over 7.3 million similarity scores.
But how do you even begin to prove that a similarity score of, say, 0.8 actually means anything in a physical hospital room?
By establishing a ground truth for drug response. For the human patients, they looked at actual clinical records. They tracked endpoints like overall survival and progression-free survival to see if a patient was sensitive or resistant to a specific drug in real life.
Right. Did the patient live and did the tumor shrink?
Exactly. And for the cell lines, they looked at the IC50 value, which is just the concentration of a drug required to kill off 50% of the cells in the lab dish.
So they matched the medical charts against the lab results. Did drug X save the patient? Did drug X kill the cell line? The logic being that if the patient and the cell line share a very high similarity score from the algorithm, their physical reaction to the exact same drug, should be identical.
The researchers quantified this by looking at widely used FDA-approved therapies. Drugs like gemcitabine for bladder cancer or erlotinib for lung cancer. And what they found was that as the similarity score generated by CTD-PathSim 2.0 increased, the matching percentage of the actual drug response consistently skyrocketed.
That's huge.
It is. The tool heavily dominated the older single-omics methods across the board. It achieved the highest weighted macro-averaged concordant scores.
But wait, is a computer really that trustworthy? When dealing with machine learning models classifying cancer types, I saw the paper mention they hit a macro-averaged F1 score of 0.96. And for anyone listening who isn't a data scientist, an F1 score basically tells us how good a tool is at finding the right matches without accidentally setting off a bunch of false alarms.
Yeah.
A score of 0.96 is basically perfection.
It's extremely high.
So how do we know the algorithm isn't just hallucinating a match to make its own math look good? AI hallucinates all the time.
And that skepticism is exactly why one specific finding in their clustering analysis is so vital. When the algorithm tried to cluster and classify a subtype called HER2-enriched breast cancer, it didn't give a 0.96. It gave an F1 score of NA, not applicable.
At first glance, that looks like the algorithm just crashed. It threw its hands up and classified 67% of those samples as luminal A and 22% as basal-like. It completely failed to put HER2 enriched into a neat little box.
But that NA is actually a massive victory for the model's integrity. It proves it isn't hallucinating to please us.
How so?
Because clinical literature has recently proven that HER2-enriched tumors don't have a single transcriptional identity. They are famously wildly heterogeneous. In real patients, they frequently show up exhibiting luminal A or basal-like characteristics. The algorithm refused to lie. It recognized the underlying biological chaos and highlighted it, rather than forcing the data into a clean, false categorization.
It looked at a messy biological reality and said, I can't put this in a neat box because biology didn't put this in a neat box. That level of nuance is incredible. It honestly makes the high scores in the other areas so much more believable.
Which really points to the broader implications of this entire methodology. If we connect this to the bigger picture we've spent this entire time talking about oncology and tumor mutations, but mathematical unblending, this deconvoluting of multi-omics data to find a perfect proxy, it isn't limited to cancer research at all.
Right. The researchers explicitly highlight other diseases in the paper. They mention diabetes specifically.
The exact same pipeline can be applied there. You could take the complex multi-omics profile of an individual diabetic patient and match them to specific diabetes cell lines, like the MIN6 pancreatic beta cell line,
which is heavily used to study insulin production.
Exactly. You could verify mathematically that the MIN6 cells actually represent that specific patient's genetic and epigenetic profile before you ever begin testing a new experimental insulin therapy on the cells. You are ensuring the proxy is flawless no matter what disease you are trying to cure.
Because if the proxy is perfect, the clinical trial is vastly safer, faster, and way more likely to succeed.
Precisely. It changes the whole game.
Which leaves us with a truly mind-bending concept to mull over as we wrap up. So if you're listening, think about this. We started this whole conversation talking about the terrifying gap between the plastic lab dish and the human body, the tragic reality of a cure that only works in a vacuum. But if algorithms like CTD-PathSim 2.0 can mathematically align your exact, messy, highly individual biology to a lab-grown proxy with this level of pathway accuracy, are we approaching a new era of medicine?
It really feels like we're on the edge of it.
Imagine a future where your specific disease is mapped, fought, tested, and entirely cured in a petri dish before you ever swallow a single experimental pill. They aren't just testing a drug anymore. They are testing it on a digital biological twin of your exact tumor.
It's an incredible thought.
It really is. Thank you for exploring this fascinating shift in computational medicine with us today. We'll catch you on the next deep dive.